Hello student, today we will start the structure of ovum. All eggs are surrounded by a cell membrane known as plasma lemma. With a few exceptions, the egg is enveloped by an additional membrane produced in the ovary, which is known as the vitelline membrane. The vitelline membrane in mammal is known as the zona pellucida. Secondary membrane secreted by the ducts such as the chorion of insects or the jelly coats of the amphibian. Eggs also occur, but they are less important. The cytoplasm of the egg contains various organelles, yolk granules, and often large amount of pigments. A well-defined layer of gelated cytoplasm, approximately 2 to 3 microns in thickness, is present below the cell membrane and is termed as cortex or cortical layer of the egg. The cortex contains granules of the mucopolysaccharides. Nature of a size range of 0 to 9 to 1 to 5 micron approximately. The cortex may also contain varying amount of pigment while the egg is still in the contact with the follicular cells. Follicular processes may penetrate the cortical layer. This penetration may be of importance in determining the specific character of the cortical layer in some animals. Polarity of eggs. What is meant by polarity? Some organization with the egg is identification indicated by its polarity. All animals' eggs have the polar structure. All animal eggs have the polar structure. The two opposite poles that can be distinguished are referred to as the animal and vegetal poles. Animal and vegetal poles. Upper pole, anterior side, that is the animal pole, and lower means posterior side pole is the vegetal poles of the egg. The imaginary line connecting the pole defines the main axis of the egg and is known as the animal vegetal axis. The polarity of the egg is often apparent in the arrangement of its cytoplasmic inclusions in many eggs. The yolk is not evenly distributed. Often its density increases from the animal pole towards the vegetal pole with corresponding the structure changes. In some cases, there are subsequently differences in the degree of pigmentation. In many amphibians, the animal region of the egg surface is heavily pigmented, whereas the vegetal region is essentially unpigmented. Various theories have been suggested in an endeavor to explain polarity in the fully developed egg or oocyte. All these theories emphasize qualitative and quantitative differences in the cytoplasmic substance. extending from the one pole of the egg to the other. The animals and vegetal poles of the egg have definite relationship to the organization of the chordate egg. In Amphioxus, the animal pole becomes ventro-anterior part of the embryo, while in the frog, animal pole area becomes cephalic end of the future tadpole, and the yolk pole come to occupy the posterior aspect. In telost and elasmobranch fishes, the yolk-laden poles lies in the future ventral aspect of the embryo, and it occupies a similar position in the reptiles, birds, and prototherian mammals. It has been observed by several workers that the various substances in mature vertebrate and protochordate egg tends to assume many animal egg possesses a bilateral symmetry. It may be expressed in the shape of unfertilized egg, as in most insects. Bilateral symmetry in many vertebrate and protochordate eggs appears when the fertilization